Hello everyone, I hope you are right wherever you are, and I'm very sure that we are still going on very well as I expected. As I had promised you earlier that in our third lesson we are going to look at the temporary slide that is prepared from the onion bulb. I'm going to carry out some few stages that will enable us prepare the temporary slide for an onion epidermis. Remember, there are two types of slide. There is the temporary slide and the permanent slide. Temporary slide is the one that is used once and discarded, while the permanent slide is used and it can be stored for future, then it is reused in that process. So I would like us to start checking on the apparatus that we are supposed to have during this process when we are preparing a temporary onion epidermal cells. The first one is the microscope. We are supposed to have the microscope here, which will be basically used for magnification. We are also supposed to have an onion bulb. There we go. I hope you are able to see it very clearly. We are supposed to have a scalpel or a razor blade if you may not be able to find a scalpel. We are supposed to have a clean glass slide. I hope you are able to see this clean glass slide over here. We are also supposed to have this one here. I call it the cover slip. It contains some droplets of water. I hope you are able to see it right there. We are also supposed to be having a dropper. This is a dropper that we will use, but later on. We are also supposed to be having some distilled water and the iodine solution. I hope you are able to see the distilled water is here. The iodine solution is also down there. I would like us to start the procedure one on one as we continue to move on so that we can now view our onion epidermal cell. Procedure number one, cut the onion bulb vertically. Therefore, in some books it is written longitudinally. A longitudinal section means from up to bottom. Therefore, I'm cutting it. I think you're able to see the way I'm cutting my onion bulb. I'm cutting it vertically. After cutting it vertically, I chop it down from this part and from this other part as well. Then I remove a small strip. Inside this strip, there is the outer part. There is a part containing the epidermal cells. Remember, we say that the epidermis protects the inner part of the tissue. When cutting this part, I must use a sharp blade to ensure that I don't denature the cell. That is to avoid denaturation of the cells. Upon cutting, I now go to procedure number 2, whereby I'm going to remove the epidermis from the onion bulb. There we go. I told you that the epidermis is found inside here, and I'm going to remove it very easily. I'm placing a mark where it will cut at. I'm also placing another mark down here. I think you're able to see what I've just done. I'm supposed to remove epidermis from in here. And remember, upon removing this epidermis, I'm supposed to place it on a glass slide which contains some water. Therefore, it means that I need to place at least a drop of water on the glass slide so that I just place the epidermis on it. There we go. This is my distilled water. I only need some few drops, one or so. Then this is some water that I'm going to place into my glass slide. I hope you are able to see that very well. And just a drop is enough. Let me now remove the epidermis and place it onto the glass. Hope you are able to see what I'm seeing very closely. Let me just try it out. I think you're seeing what I'm doing. There we go. Okay, it is snipping off, but that is okay. As long as I get a small piece, This one here is an onion epidermis which contains some cells in it. I'm now supposed to place it here by use of a mounting needle, but I'm just going to improvise so that I place it using this dropper. When I'm placing it here, I must ensure that there is no bubbles being formed on the slide, because in case there are some bubbles, then it may end up not being clear enough. The bubbles may form some dark structures. You can now place your cover slip. I think you can see my cover slip. It's on top here. I've already placed it on top of the specimen that I've already prepared. Then I now move it gently to the stage. Remember, the reason as to why I placed a drop of water here is to make the cells turgid. There we go. I'm placing it here so that I can try and view it. There we go. Yes, I think my cells are now visible. Let me just make it visible so that you can also be able to see it. It's right. I can now clip them up there. Yeah. Let me do it once again. Right, there we go. You can now view my cells that I've just placed here. Just view my cells. Right, there we go. These are the cells that I just prepared. You can just be able to view them. I hope you can see them now. Hope you are going to see them. There we go. That's the bright field of view, which contains some cells in it. I think you can see the cells up there. Right. Yeah. I hope you are able to see my cells now. Those are the cells. You can count them. 1, 2, 3, 4. They are 4 cells along the field of view. Therefore, upon doing that, we can now proceed on. We can also place the iodine solution so as to stain the cells. Remember, for the first one, we only placed a drop of water which made the cells turgid, but they were less visible, and that is why we strained while trying to look at them so that they become so clear. And therefore, right now, we are going to use the same epidermis that we prepared, but now we are going to place some iodine solutions instead of water. There we go. I'm picking goes out. Capture kabisa. Can you play near me? Right, then you placed some few drops of iodine. I think you have seen the drops of iodine that I've just placed there. Then we can go to the microscope and view the specimen because it's now ready for viewing. Right, perfect. I hope you'll be able to see this as I've just seen it. Even the nucleus is visible. Now you can still move closer. Just move closer. Let me just try to view it so that we can see it. Let me just try and see if we can be able to view it. I think it's visible. My cameraman is doing some great job. Yes, you can see the cells down there. Even the light microscope, the nucleus is visible. You are also able to see the cell wall in addition to cell membrane. When we use the iodine solution, it becomes much visible, and therefore it becomes so easy for an individual to be able to notice even small structures that couldn't have been seen by the use of a drop of water only. Iodine is used for staining for clear visibility. That is a common question. You're asked to state the reason as to why we use the reason as to why iodine is better compared to water is because it brings in clear visibility. That marks the end of our third lesson concerning preparation of a temporary slide. But don't go away, because in the next lesson we are also going to look at another aspect. That is, in the fourth lesson, we are going to look at how to estimate the cell size. Remember to subscribe, and we are still going to learn more. Thank you.